you very much for inviting me. Okay, so my title today is Novel Mechanism of Adjuvanticity Implied from In Vitro Studies with Bacterial Toxins. Now, most of us have heard of cholera toxin and Vibrio cholera that causes diarrhea in humans. Now, this toxin, or this family of toxin, E. coli heat label enterotoxin, they're very similar to cholera toxin in the function and the structure, although they have also unique properties. Now, nature have delivered us these nasty toxins, but at the same time, they are wonderful molecules. If we look at the structure of E. coli heat label enterotoxin, we have what we call toxin 1 and toxin 2. LT1 or toxin 1 okay. Is it this one? No. The laser.
It's the one in the middle.
In the middle. Okay, great. So if we look at the crystal structure of both toxins, there is the upper part, which is the A1 subunit, and A2 subunit. The A1 subunit catalyzes ADP ribosylation. It causes ADP ribosylation, which is eventually lead to the imbalance in electrolyte and diarrhea. Whereas the A2 subunit, which is linked to A1 subunit, and spins or goes inside a donut shape part of the toxin, which we call the B subunit of the toxin. Now, this donut shape of the toxin is completely non-toxic. The interesting part of this toxin is the compact structure of it. Because of that compact structure, it allows or it confers high stability to the toxin, both at temperatures over 60 degrees, as well as in pH as low as 2. So the other thing, what is interesting about this molecule is that it's a pentamer. It's formed from 5 identical monomers. Each monomer can accommodate a protein up to 40 kilodalton. So that's a big protein relatively. So what this means, one can conjugate 5 proteins to one subunit, allowing it to basically deliver this protein to the immune system. Now, how does it deliver it? It delivers it because it binds very effectively to a receptor. It's a ganglioside receptor. It's called GM1 receptor. And this receptor is a glycolipid receptor, basically form of sugar, sialic acid, galactose, and glucose, and has a ceramide tail, which is inserted into the membrane. Now, this, the binding or the affinity of binding is pretty high. And people have exploited the binding to the receptor to deliver proteins into the cell, as well as we're going to see in a moment, adjuvanting immune responses to other proteins. Now, LT2 has similar structure as we see it here to LT1. However, in terms of amino acid sequences, they differ. The interesting part of our LT2 toxins is that it binds to a complex of receptors. It binds to another ganglioside, GD1B, which has basically an additional sialic acid to GM1, plus the TR2 receptor. That has been shown by FRET analysis and fluorescence microscopy. So the importance of binding to these receptors, we're going to see later on, is exemplified in their effects on the on immune cells. Now, some work we did earlier in the UK with Tim Hurst. He was in the field of bacterial toxins. We actually published what we thought and what others thought a landmark study where we prove the importance of the receptor binding. So what we generated a mutation, which is one single mutation, glycine to aspartic acid. And with this single mutation, we can knock out, actually, we can eliminate binding of the wild-type protein. And if we inject both either the wild-type or the mutant, you can see that you could generate a very good IgG response in mice with the wild-type, whereas very little or non-existent immune response, antibody response to the mutant protein. We can also see this if the toxins are delivered by the oral route. We can also generate an antibody response, IgG and IgA in secretions. So this established, in a way, the importance of binding to the receptor in, at least at this case, generating an immune response. We also looked what happened if you take just the spleen incubated with these toxins. So there's no immunization here. This is in vitro. What happens if you incubate it with the wild type and with the mutant? And we can see there is upregulation of co-stimulatory molecules. These molecules like class 2, B7-2, ICAM1, CD40, etc. So the simple incubation of mouse spleen cells with this toxin can upregulate co-stimulatory molecules. We also look at cytokine production, and we can see also, if we compare the mutant to the wild-type toxin, and here what we're looking at is CD11C positive cells, dendritic cells. We can induce the proliferation of T cells with the wild type, whereas little proliferation is induced with the mutant. So these were mice that were injected either with the wild type or the mutant protein, and then the spleen was taken and in vitro, proliferation was measured. We also can see it generate massive IL-4 and interferon gamma, whereas the mutant much less significantly. So this established in a way the importance of receptor binding to generating an antibody response. We went also we reasoned also that if it has such a powerful effect, why not testing it in disease models? And we could demonstrate that the toxin 1, at least, that it protects against CIA. So what happens here, it can protect against collagen-induced arthritis before the disease is established, which is usually after injection of collagen in adjuvant, or even just before the signs are manifested. So it can protect before or after. And you can see here a cross-section of the joint cartilage, the destruction of the lining, whereas in the case of administration of LT-IIaB, the lining is perfect. Infiltration of cells in the case of non-treated cells, and in case of treated cells with LT-IIaB, little infiltration of cells. So this toxin seems to have powerful immune modulation effects. So if it does this, does it do it for other antigens which are administered with it? And we can, what we did here is, again, we conjugated OVA, and we conjugated the mutant protein or OVA, and we tested also the medium. We used the system here of A20 wild-type B cells and the OVA-responsive T cells, DO11. So we can see here also that the wild-type toxin at different doses can induce a good proliferative response. In the case of the mutant, is almost non-existent. So this means that the toxin can deliver antigen into the cell and can generate a proliferative response. In fact, a number of genetic fusion proteins have been made by other investigators, or they simply administered this protein with LT-IIaB, and in all cases, they could generate a good immune response, and they could protect against the pathogen from which these proteins are derived. So what happened with LT-IIaB, the other toxin? The other toxin also can stimulate expression of co-stimulatory molecule. It can stimulate those molecules here, CD40, 80, 86, etc. And in this case, what we have, the APC is CD11C, and the proteins, either OVA or OVA plus the toxin, were administered intranasally. The other toxin, or type 2 toxin, also have a number of properties. It increases secretion of inflammatory monocytes, cytokines. It increases activation of NF-kappa B, induces migration of DCs by CCR7, proliferation, and antibody response to co-administered antigens. So both the proteins seem to be working similarly. However, the main difference between them is in the case of the toxin 1, it induces a Th2 type of response, whereas the toxin 2 induces a pro-inflammatory cytokine response. So we've been interested since we investigated the mechanism of these toxins, whether other mechanisms exist. And for that reason, the reason we thought that there could be other mechanisms beside binding to the receptor and inducing cytokines and antibody responses is the dose. It is sufficient to administer with another protein or on their own 1 to 50 nanograms of the toxin, or 35 to 30 micrograms of them in the absence of any adjuvant. That's in PBS. So you can induce adjuvanticity with such a very small molecule, such a small dose. So the question is here, because the receptors are expressed ubiquitously on mammalian cells, how could sufficient dose of the toxin reach the APC? For example, in the case of LT-IIaB, it binds not only to B cell or dendritic cell or macrophages, it can bind to T cells, it can bind to epithelium, it can bind to endothelium. So how a very small dose can induce such a good response? If we look at the immune response, not only very high antibody response and high level of cytokine, but also a memory response up to 2 years. So that's extensive. What this means that this molecule can induce a long-term immune memory response, not only to themselves, but also to other antigen administered with them. So our approach was to look at other things early during incubation with the toxin. So while we look at whether there is interaction between T and B cells, or whether the toxin does anything to T and B cell early during incubation. We look at T cell division, IL-2 production, B cell antigen presentation. All of these we know they are important in the immune response to an antigen. Now, what is interesting about, for example, in this case, toxin 1, if you look and incubated with Jurkat T cells, you see the distribution of the toxin on the surface of this cell. Now, remember that GM1 is a part of lipid rafts. And GM1 and lipid rafts, they are those microdomains in the membrane that, that have signaling molecule. They have protein which are linked to signaling molecule. So any changes on the membrane will result in signals that would consequently affect, if you want, how these cells behave. And if we look here, within 5 minutes, we can see the distribution of the toxin on the surface of the cell. Well, if you wait a little bit, up to 15, 30 minutes, you see that capping occurring on the membrane. So that capping, meaning what you have here is several GM1 molecule or rafts pulled together to one pole of the cell. So that has to have an effect on how the cell behaves in terms of signals that are delivered to the cell. If we incubate these cells with the mutant protein, we see obviously no binding and no capping. The other things is what we looked at is whether, because of that high affinity binding and because of capping, whether the toxins are retained on the membrane. And here again with toxin 1. What we use here is a system which I will describe later on as well, which involve a 20 B cell, but also a 20 B cell which are transfected with a human IgM. Now, what happened that the human IgM is specific to phosphorylcholine. So we can deliver a protein through the B cell receptor attached to phosphorylcholine. For example, in this case, PC ova. And we can compare these to protein which are linked chemically to toxin 1, which is sometimes called 8xB. So as we see here, that in the case of B cell receptor endocytosis, that by 120 minutes, the protein, which is ova, disappears from the surface. Whereas in the case of 8xB ova, it will take much longer time. 240 minutes, still not there. It will take 24 hours until it disappears completely from the surface. There is something happening on the membrane which retains this toxin on the surface. So we want to look here whether a short incubation can do something to the cells. So we incubated the B cells, again, A20 B cells. We incubate them with the toxin 1, toxin 2. And remember that these are non-toxic molecules. I refer to toxin, I refer to the B subunit, the non-toxic part of the toxin. But for ease, I call them toxin 1 and toxin 2. And what we see here, that short incubation, after 4 hours, we can induce clusters of cells. So those are B cells, which are clustering. With toxin 2, even larger clusters can be induced. And when mixed with toxin 1 and toxin 2, we can also induce a mix of larger number and larger size of a cluster. Now, why this is important? Because it's known to induce a good immune response that the interaction or the clusters that are formed, the number of clusters and the size of cluster that are formed between APC and T cell are very important. So we went further and look here, what would happen if you add T cells? Or actually before that, we wanted to see whether the receptor has to do anything with cluster formation in B cells. So a property of these toxins, if you heat them to 95 degrees Celsius, they disassemble, so they become monomers. And it takes more than one monomer to bind to the receptor. So that's a nice way of looking at the importance of the receptor. So we compare unheated to heated sample, and we can show with unheated samples, the clusters are found, and with the heated samples, there are less significantly number of cluster and size of cluster which are formed. So this in a way says that the toxin can bind to the receptor and form the cluster. Now, what receptor is involved? We thought about the ganglioside receptor to be the main important receptor, but remember toxin 2 also binds to TR2. So we generated a mutant that does not, of the same toxin, which does not bind to TR2. And in this case, but still retains binding to ganglioside. So what happened here is that after incubation of this toxin, we can see that there are clusters in both the wild type and the mutant protein. So this says that gangliosides are mainly involved in formation of these clusters. We went then further. We added, we wanted to mimic, and we wanted to mimic the antigen presentation assays. And so what we did here, we CFSE labeled T cells, and then we add them to B cells. Again, those B cells are I20, wild-type B cell, and the T cell are DO11. So we incubate these toxins in the presence or absence of ovalbumin. We could see that the clusters are formed again, and we could see the interaction of these T cells with the B cells in the culture. We can see it here after 24 hours, also that those T cells interact with the B cells or are present within these B cells in these clusters. So in a way, this tells us that these cells, the binding to the receptor is important in formation of this cluster, and there are interaction, at least under the microscope, between B and T cells. Viability of the cells is maintained, irrespective. So that's not, for example, dead cells which release DNA and causes this clustering. Now, we went further to look at antigen presentation. We use here the same system. We use the A20 wild type, and we use the over-responsive T cells. So what we did here, what we're looking here, is at cell division. So these T cells have been labeled with CFSE, and in antigen-presenting assays, where we can put the ova alone, we put the toxin alone, or we mix the toxin with ovalbumin. And what we could see, the take-home message from this, is that when we looking at T cell division, which progress from 0 to 7, so we look at 7 generation here, what happened to the T cells, is that in the presence of the toxin 2, there is acceleration, whereas in the case of toxin 1, there is a delay in T cell division. So this is using what? Is using ovalbumin fluid phase endocytosis. So these cells do not, the B cells do not have a receptor that can recognize a known antigen. So the ova has to be internalized by fluid phase endocytosis, or by fluid phase. Now we looked at B cell receptor endocytosis. And in this case, we use the same system, but the B cell, remember, have been transfected with human IgM, which is specific to phosphorylcholine. So if we deliver now B cell PC ova, and we do the same thing, incubate the cell with the toxin, very surprisingly, that most of the cells are stuck at 0 generation. So what is happening here, it looks like the toxin in the presence of ova is stopping the cells to divide. So the next question was, are these T cells functional? And they are functional. They're producing IL-2. And they're producing IL-2, particularly the toxin 1. Toxin 2 also producing IL-2 during the fluid phase endocytosis. But during BCR-mediated endocytosis, toxin 2 does not seem to produce significant amount of IL-2. And again here, well, we see LT-IIaB with the PC ova producing large amount of IL-2, and a mix also producing large amount of IL-2. And finally, what we did here is to look at antigen presentation, but after different times. So those APCs were fixed, and then we looked at T cell proliferation. The T cell proliferation, we looked at HT2 T cells, which are responsive to IL-2. And what we can see here with the ova, when it is targeted to the B cell receptor, within 30 minutes, it starts producing IL-2. And that level of about 240 minutes. Whereas in the case of the toxin 1 of albumin, well, we see it takes up to 120 minutes to do anything, and you only start at 240 minutes, and it is increasing afterwards. So there is a delay in antigen presentation with these, in the presence of these toxins. Now, so the final slide, which is the interpretation, how do we have what we see an inverse relationship between T cell division and IL-2 production. That's not surprising. T cell hybrids, they stop dividing when you challenge them with antigen. And they start producing IL-2. That's due to a block in the G and S phase. Temporary block, which is usually for 48 hours or so. But what is interesting here is that the toxins themselves are doing the same thing alone in the absence of ovalbumin. So what this means is that in the case of these toxins, they're blocking the T cell division, and they're producing IL-2. So they're mimicking what an antigen does through the B cell receptor. Now, in the case of LT-IIaB, we think that in this case, if the B cell receptor is delivering a signal through the B cell receptor antigen, and there is concurrent signal from GM1 receptor, this interact or this affect the cytoskeleton. And the cytoskeleton, by affecting the cytoskeleton, it increases adhesiveness of the cells. So we know that increase in or movement of the cytoskeleton can affect expression of adhesion molecule. So we think the early, if you want, the early adhesiveness or the early clustering that we're seeing is due to the effect of the toxin binding to GM1, affecting the cytoskeleton and affecting, as a result, the molecule, adhesion molecule. In the case of IL-2, well, in this case, there is IL-2 production. But in the case of LT-IIb, there is no IL-2 production. And the reason, because here we have 3 receptors, and 3 signals at the same time are delivered to the cell. And as a result, there is a blockade in IL-2 production. Okay, I'd like to thank my students, PhD student, Siham Al-Qassas, Rawa Faraj, and Karmaka Macha Martin, Tuskegee University, collaborator Terry Connell, University of Buffalo, George Haji Shingali's Pennsylvania School, and Jim Drake at Albany Medical College. Thank you.